Hello, my name is Professor Matthew Schmidt, and I'd like to welcome you to genetics. Today's session deals with the concept of sex linkage and sex-linked traits. I purposely put it where I did because we've already spoken about the chromosomes in general, how they move during meiosis and mitosis, and their role in sex determination. And while this is not 100% linked, as it were, to that topic, it does have to do with integrating Mendel's principles with the fact that there are different types of chromosomes. So let's go ahead and see. By the way, Mendel knew nothing about sex linkage. When it was discovered, it was thought that it might be an actual exception to Mendel's rules, but it turns out that Mendel's rules always work. Sometimes you just have to figure out what is going on that's a little bit different than what he saw with his pea plants. So let's figure that out. So first of all, the term sex linkage and sex-linked traits, what it really means is that some genes, and the genes rarely have anything to do with sex at all, happen to be located on the X chromosome. Now, just referring to the last session, we established, remember, that at least in humans, the X chromosome really doesn't have a lot to do with determining whether someone's male or female. That is, even though it's part of the sex chromosome pair, the X really is a regular chromosome with tons of genes on them, and those can be genes that have to do with anything. So if they happen to be located on the X, they are sex-linked traits, and in fact, often this term sex-linked, while extremely important and valuable, if it means the genes on the X chromosome, often you'll hear it referred to as simply X-linked. It's sort of a coincidence that sex and X rhyme. They sound very similar, but the thing is, if the gene's on the X chromosome, it doesn't affect its functioning per se, but it does affect potentially the results of Punnett square type crosses. Then we would see something that Mendel would have probably been a little confused about if he had done this. So, as I say here, Mendelian laws still hold, but the results of a typical cross, while they look, they look somewhat similar to what you're used to, they may be modified. Now, one thing we should be very clear about here is Y linkage is possible, but it's exceedingly rare, and almost always normally when people talk of sex linkage, they really mean X linkage. Be a little careful though, because in some problems, which we'll see as we go forth, they do ask you to consider the possibility of Y linkage. It is possible, and there are certain pedigrees, which we'll get to in the next session, where Y linkage might be the only possible explanation for the data that you see. But generally speaking, Y linkage is not what people mean when they talk about sex linkage. And certainly when it was discovered, it was a gene that was on the X chromosome. Now, I want to give you some examples in Drosophila. And the reason is, first of all, sex linkage was discovered in Drosophila. And when it was discovered, as I mentioned to you before, it was thought that this was really something revolutionary. And it was important, but in the sense that they thought maybe this was an exception to Mendel's principles. It turns out, as I've said, and we'll see, that Mendel's principles always work. We just have to take something else into account here. So let me give you a little background information about Drosophila, one of its important traits, and this really, I'm pretty sure, was the first X-linked gene ever discovered that we can work with here. So in Drosophila, remember, don't worry right now about the fact that the sex determination system works a little bit differently. There's still an X chromosome and there's a Y chromosome, and the genes that are on the X really don't have per se necessarily to do with sex. So in Drosophila, in the fruit fly, the normal eye color is a bright red. If you go out and look at some fruit flies at your local fruit stand, if you can see close enough, you'll see that they do have red eyes. That's the normal or wild type condition, which is a term we'll discuss a little further in a moment. Now it turns out, we now know this, they didn't know it when they discovered it, but what they discovered is that this eye color gene is located on the X chromosome. And just for the record, probably the reason that it was discovered in Drosophila, and in fact the reason why there are a lot of X-linked genes in Drosophila, is because there are fewer chromosomes. In the pea plant, remember we said there are 50 chromosomes with n equal to 25. In Drosophila, n equals 4. So that implies if all the chromosomes are about the same size, that fully 1/4 of the genes in the Drosophila genome will be located on the X chromosome. So you'll see a lot more X linkage in Drosophila than you would either in humans or in pea plants. So it makes sense that it was discovered there. Now, a mutation in the eye color gene causes the fruit fly to have white eyes. It's a lack of pigmentation. You can see it very easily and readily under a microscope. You could really see it. Sometimes if you look close at a regular fruit fly, you'll see it has white eyes, but generally not out in the wild because this is a mutation that occurred. It was rare, but it's been propagated over the years so that we have a lot of white-eyed flies in the laboratory, but not so much out in nature. Now, they're a little bit different terminology that shouldn't get confusing, but Drosophila geneticists use slightly different conventions than Mendel did. So firstly, the gene was discovered that caused white eyes, so they named it little w. It's little w for white because they then realized that this little w was recessive, in fact, to the normal gene that caused red eyes. So remember what Mendel did. He named the trait based on its dominant form, like purple flowers was a big P, and then white was a little p because it was recessive. Drosophila genes mutations tend to be named after the mutation. It doesn't have to be recessive, but often it is. So that's what happened in this case. So it's important to know, and you don't have to memorize it, although it wouldn't be a bad idea, but usually in a problem they will tell you which gene is recessive and which is dominant. Now, the normal gene, therefore, here's where the terminology gets a little bit different. You can call it capital W. That would be the Mendelian way to do it, right, in a sense. But usually what that gene is called is little w plus. The plus refers to the so-called wild type, the term I mentioned earlier. Wild type means that this is really the normal situation. Remember, in Mendel's peas, you couldn't say it's better or more normal to have purple flowers or white flowers. They were just two variants that you found out in nature. This mutation is a very definite, somewhat rare thing that happened. So in the bulk of natural fruit flies, you don't see this trait. So wild type is, go out and look at them. You'll find that they all have red eyes. But it's called W plus because remember the gene was named after the recessive allele that caused white eyes. Very often, the abbreviation just plus is used. You don't even say the W. We'll see that as we go through some problems. It depends, as long as you've established what the gene is that you're talking about and you want to do problems and save a little time and do some shorthand, plus is just as good as saying something like W plus in this situation. So take a minute to take that all in. As we go through it, I think you'll get comfortable with it. So the bottom line is this. If Mendelian principles still hold, if we're going to do a cross involving this gene, we have to not only keep track of the alleles, which is what a normal Punnett square does, but we also have to keep track of the chromosomes. So it's a little bit more complicated, but please keep in your mind the exact same principles are happening here. It's just different because when a gene happens to be on the X, as we're going to see, there's differences between the two sexes because females have two X's and males have an X and a Y. So first of all, let's consider what the possibilities are. If you're female, first of all, perhaps obviously, you're going to have two X chromosomes, right? That's something that we've been leading into here that we've talked about in the last lecture or the last topic. And males are going to be XY. You may say, yeah, we know. But if an allele, a gene, is located